good morning or good evening, depending on the time you guys watching this video. My name is Badi Ibrahim. I'm a medical doctor working in the field of neurology. I'm glad here to be able to participate in the IHS Summit for 2022, to be able to present our topic for today about space neuroscience, our current understanding and future research. So we're going to talk briefly into our outline for today, and then we're going to sail into our presentation. So starting with our outline, we're going to talk about brief introduction, effects of space travel on the human brain, potential areas of research in space neuroscience, points of bridging the gap between theory and practice, and going to end up by exclusion. So starting with space exploration, as we all know, space exploration is crucial for our understanding of our surroundings, and to establishing scientific concepts to explore, monitor, and save our planet's future. Also, exploring space will enhance our knowledge in several fields, including medical sciences, opening new avenues for technical, medical, employment, economic, and development of our healthcare system. Also, making long trip space flights and living in another celestial environment will impose by itself a lot of challenges, which hopefully all of us working together will be able to defeat those challenges. So starting talking about the human brain, as most of us know, it's a complex structure. It's used to, it has been working in 1G environment. It's a very sophisticated system, can be affected by all kinds of stressors, and definitely it has several changes under 0G forces. So our main aim of this study is to review several aspects that are related to the nervous system in wetness conditions, as well as into development of future emerging field of neuroscience. So now we're going to start to mention some of the effects of space travel on the human brain. We're going to start talking about several points, skim through it. So first of all, we're going to talk about the intracranial fluid dynamics. So basically at Earth or at 1G force, the human body has been working under the 1G circumstances that creates a hydrostatic gradient. This hydrostatic gradient pressure increases from head to toe progressively while standing or sitting, making the body fluids distribution goes from up to the toes. However, loss of 1G force in space will make this pressure not existed, making the fluid dynamics shifted upward into the cranial or into the head. And this collection of fluid in the headward, upward direction will cause a persistent increase in the intracranial pressure. So there was a syndrome not to be happening with this change in the dynamics of fluid called space flight associated neuroocular syndrome, where unilateral and bilateral optic disc edema were noted, globe flattening, retinal and choroidal folds, refractive errors, shifts, and areas of focal ischemia even in the retina were noticed. Another point of the CNS or central nervous system is altered pituitary gland anatomy. So fluid shifts affecting the anatomy integrity of the pituitary gland were noticed, especially by a study done by Kramer and his colleagues, where they conducted a prospective MRI study of 11 astronauts. They did a brain MRI study before traveling to the ISS for the astronaut, and after they returned. They have found that the brain and CSF fluid volumes had expanded during this space flight, and they attributed it to increased intracranial pressure. At 1 year post-flight, they repeated the MRI scans, where it showed that the volume increase was still there, suggesting a permanent change in the brain structure. In addition to that, they also noticed that the dome of the pituitary gland, which was predominantly convex prior to flight or prior to exposure to microgravity, were changed into flattening or convexity post-flight. The next point is going to be about the gray and white matter changes, where there are a couple of studies done showing cortical gray matter shrinkage along with cerebrospinal fluid volume. In addition to that, there was another study done on 18 subjects in stimulated microgravity using head tilt model 6 degrees for 30 days and using brain MRI scans, which revealed loss of gray matter volumes in the frontal and parietal lobes after the space flight. These changes in these locations are known to be associated with defects in the locomotion, cognition, coordination, learning and memory, and also auditory process. All of these changes that were noticed in these studies had led to the concept of space brain, which represent the physical damage to the brain caused by the multiplicity of the adverse effect after exposure to the environment of space. Another effect of zero-G can be noticed on the vestibular system, which is a multi-level structural and functional changes to alter the gravity. There was an inner ear physiological movement in non-gravitational environment loss were noticed, leading to a phenomenon called space sickness. So basically, the vestibular system is at risk as the semicircular canals, utricle, and saculae are affected by an altered body position. The inner ear, similar to the rest of the brain, has evolved over the past millennia to function at 1G. The minute crystals in the otolith organ, they move in a very thin gel. And this movement becomes non-physiological in a non-gravitational environment, leading to symptoms like dizziness and spatial disorientation, which was termed under the terminology of space sickness. Another point there was a study done on telomere, and a dysfunction was noticed. So to start with, telomeres they are the DNA cap that is found at the end of the chromosomes, and they are implicated in cell division, aging, and cell death. There was an elongation during space flight noticed at the telomere and shortening below baseline upon returning to Earth. In addition to that, an alteration in the DNA sequence was also noticed. However, the study that mentioned these changes were done on a small sample size, and it remains uncertain whether these effects were due to physiological stress or radiation. Nevertheless, it was the first reported study that has reported spaceflight telomere dysfunction. And there are different effects on different tissues remains unclear, whereas if spaceflight can have another effect. So that's another area for us to make a lot of research on. Moving to the next point, which is about sympathetic nervous system. So researchers have demonstrated that the sympathetic nervous system also suffers from a variety of effects during spaceflight and even upon return to Earth. These include increased in the heart rate, reduced in the arterial pressure, and even an experience of a pre-syncopal attack. There was actually one study that examined the influence of microgravity on the Valsalva maneuver to test the hypothesis of impaired sympathetic and vagal cardiac barrel-reflexes in astronauts. So this study showed that the Valsalva maneuver was found to elicit a larger increase in the muscle sympathetic nerve activity in space than on Earth. Also, it was found to produce a deeper fall in the arterial pressure. As a result, these findings, sympathetic action to lower diastolic pressure, known as reflex gain, was found to be normal. And this study, along with other studies, also have documented changes in the vagal baroreflex responses, all of which suggested that exposure to microgravity actually impacts the human baroreflex in a different manner, where the sympathetic baroreflex responses are intact and vagal baroreflex responses are affected. So moving to the next point, cognitive and behavioral effects of space or zero-G force. So studying cognitive and behavioral aspect of health on long distance spaceflight circumstances, especially for people going to space for a long duration of time and with the increase in the future intention to make a huge number of people travel to space, making studying the cognitive and behavioral essential to be able to choose wisely or to determine who is able or who is not to travel to space flight. So talking about that, we have established studies that have showed that people react differently to stress, in addition to stressors. And by that, having different mood disorders. However, these studies were all conducted on general population and could not be generalized to astronauts due to different demographics and circumstances. Now, with the upcoming need for researches to be done on, in order to make human or civilians traveling to space more safe trips, there is a pressing need for more researches on the behavioral and cognitive health effects of space flights, and various factors must be considered into selecting people going to space, as prolonged exposure to stress can also result in unwanted health conditions such as depression and anxiety. And there was areas of the brain that can be found, areas of the brain was found to be affected by prolonged and increased stress, also with elevated cortisol levels. For example, learning difficulties, changes in memory abilities can result from stress that can affect the hippocampus. Furthermore, mood and affective disturbances can also occur as an outcome of stress in the amygdala. So that's been done, been said, last point of the previous point. Now moving to potential areas of research in the neuroscience. We're going to start with the neuroimaging. So information out of a small number of conducted imaging studies on astronauts were done previously, but it was giving us robust information. So we need to replicate these kind of studies on a larger scale. We can do it by head of the bed down model, conducting brain MRI scans before or after defined period of times. We can monitor the fluid shift and how these changes inside the brain can affect us. So basically, these kind of studies are used by using the head of the bed down model, tilting the end of the bed 6 degrees down, and a correlation of the brain MRI scans finding and task performance is a large area of potential researches. Basically, we can stimulate the zero-G force, and we can study all kinds of effects of zero-G force on the brain, CNS, and even can be done on the whole human health system or human body. So it's actually an excellent method to find answers while we're still here on Earth. So next point, it's about neurophysiological monitoring. So cortical hyperactivity has previously been demonstrated in astronauts, and transcranial magnetic stimulation motor threshold was found to be decreased during zero-G forces, especially noticed by simulated zero-G by parabolic flight. And these changes can be seen during brief episodes of zero-G in a study done. However, the extent of which this causes functional problem to the human brain is still undetermined. Therefore, a suggestion of further studies through neurophysiological monitoring are needed for further investigation. And one potential approach could be done by analyzing the cortical neurophysiology, cortical excitability by using an electroencephalogram in prolonged head-down position. This will be enabling us to knowing different changing during the zero-G in different areas of the brain, different excitability, and different changes relating to different problems that could encounter us in the future. Another device that we can use is the pupillometry. So basically, the pupillometry is an automated device. It's an automated device, handheld, used to observe the pupillary function in a detailed manner. It has large potential uses in zero-G as being a sensitive, non-invasive tool that may detect subtle changes related to cranial-directed fluid shift, that is intracranial pressure. It functions by directing red infrared beam that detects even subtle changes that a human eye cannot detect by using the current method, using the torch. So biomarkers. Biomarkers reflecting neuronal and astrocytic cellular damage are actually a great indicator if utilized in the right way into helping us in the prognosis of certain medical conditions that can tell us the prognosis of what degree of brain injury. And association of prolonged microgravity and low dose gamma radiation with increased oxidative stress biomarkers were noted in a couple of studies. And monitoring these biomarkers in space is challenging, especially when collecting and analyzing the body fluids, such as the serum. It requires us to freeze and carry these samples and even transporting it from space to the lab. So a lot of obstacles encountering this. However, it's a large potential of tool for monitoring in space. Regarding the therapeutics point, there are very limited studies on space pharmacotherapy, and there was a trial investigation of medical and pharmacological approaches for mitigating increased intracranial pressure. But we need also to conduct even more to have a true value in the future. Observing the effects of therapeutics drug on imaging, this is I think that we can easily do. And for example, acetazolamide, if we observe the effect of it, we can make it a part of management of the increased intracranial fluid shift that we mentioned previously. And we can even send it for those who's traveling to space. So that's being done. We're going to move to the last point, bridging the gap between theory and practice. So to sum all of these things up, basically, we need a lot of research to answer the huge amount of questions that we need to answer. Especially the pathophysiological effects of the long duration space flights on the zero-G. We need a space exploration platform, making us able to deposit and share information. We can do regular meetings and workshops. Also, traveling into space, exploring has its own advantages, like establishment of valuable researches, facilities, making resources, having economic solutions, industrial expansions, tourism and employment opportunities. Also, space neuroscience research on Earth has its own value, like stimulation of analog environment to zero-G, studying of zero-G effects on Earth. So to sum up, long-distance space flights and the establishment of interplanetary space facilities is necessary for the future. Cephalic fluid shift and abnormal intracranial dynamics are the major contributors. And space neuroscience, especially, is an embryonic field that requires a significant amount of effort, dedication into the development of safe future trips into space. So by this being said, I hope you guys enjoyed, learned, or even grasped a couple of information. Hopefully it was informative topic for you guys. And thank you for letting me or giving me the opportunity to share this with you. Thank you. And these are my references. Feel free if you want to capture or read them all. And that's it. Thank you all.